Yeah, thank you. Hi. It's great to be here and be able to talk about this, as I think, exciting topic. And it actually fits quite well into the end of the last session, where this idea of how we can use AI to better understand the brain, also in terms of foundation models, could how these could facilitate research. And yeah, you can tell me at the end of the talk if this if you agree in that this might go into the direction of a foundation model. Yeah, but let's first start with a little bit of context. So Katrin has luckily already talked about quite a bit what the context is. So in the context of the Jülich Brain Atlas and the eBrains Atlas, and of course with it as part of this with BigBrain, our goal is to build a high-resolution, multimodal and multi-scale atlas of the human brain. So a three-dimensional model which combines information about different aspects of the brain, of different imaging modality in a common reference space. With CytoNet, the method that I'm presenting here, we are currently mostly operating in Cytoarchitecture, which all of you are probably familiar with. So we are looking at the organization of cells and the properties of cytoarchitecture, so columnar, laminar arrangement, the formation of brain areas, and so on. At the end of the talk, I will also comment a little bit on how this can be transferred, or how this could be transferred to other modalities as well. But for now, we focus on cytoarchitecture. So what we wanted to approach in this with this method is characterization or decoding of human brain architecture, here specifically cytoarchitecture, which means that I want to identify in general structural entities, which could mean brain areas, lamina, columns, and other structures that you could think of. So a very, very general task. And we want to define and identify these structural entities at a whole brain level and identify them in a mostly automatic way, to also address the fact that we have quite a lot of inter-individual variability. So we want to be able to, at a large scale, analyze large amounts of imaging data and map these structural entities to, at the end, associate them with functional connectivity, genetics, and molecular organization. So to give a more concrete example, one very, very prominent task in this direction is the identification of brain areas, which I'm mentioning because we will also I will also show how we evaluate the method that I'm showing here for this task. So if you're not so familiar with this, so basically the task is, given high-resolution cell body stained data, we want to look at how the cytoarchitecture is locally composed, and then based on this information derive which brain area we are looking at, and finally arrive at a segmentation or classification of this task. In this work, I developed a method which is called to do this. I developed a method which we call CytoNet, which is a model that is able to, based on large amounts of image data, automatically derive cytoarchitectonic features, which are not specifically tailored for a specific task, but are expressive enough to facilitate a wide range of different tasks, including brain area classification, segmentation of cortical layers, and maybe even other tasks. This method is based on self-supervised learning, so SSL, which is the same method that is, for example, used by large language models, in the sense that we don't tie it to a specific task, but give it a very, very generic pretext task from which it then hopefully learns to derive reasonable features of cytoarchitecture in this case. And I want to briefly, before I show you the results of this method, I want to briefly show you what the idea behind the learning paradigm is. So the key idea behind the method is that spatially close locations in the brain are likely to share cytoarchitectonic structures. Of course, you might immediately say this is not always true, for example, at cytoarchitectonic borders, but we will later see that our method is able to deal with the examples where this hypothesis is actually not true. So what we do is we take a data set of multiple brains for which we have high resolution, so typically 1 micron, 2 micron scans of histological sections, and then we bring them into a common coordinate frame to allow us to actually compute the spatial distance between different points. In this case here, we used the MNI colon 27 space, but it can actually be any space. If you're dealing with only a single subject, you can even use just the native space of this subject. So the specific space doesn't really matter. The point is that now what we can do now is for any given location in the brain, we can compute the distance to any other point in another brain through this reference space, and we can also associate each point with a point in one of our histological sections or microscopic scans of histological sections. We can then extract high-resolution images at these locations, and then we can feed these patches into a deep neural network, which can be any neural network that is suitable for processing image data. And what we then do is we basically use a so-called contrastive loss, which some of you might be familiar with, to make sure that images that are located close to each other physically in our reference space are mapped to high-dimensional feature vectors which are similar to each other. And points which are far apart are mapped to very dissimilar feature vectors. This is the loss function, so it's a variant of the so-called info-NCE loss, which is a common loss used for contrastive learning, for self-supervised learning that some of you might know. The main differentiation is here that we define a similarity on the distance really. So what we do is we take, in this case, microscopic data from 9 brains with about 1 million of these patches. We train the model, which takes quite a bit of compute, and we end up with a data set of feature vectors which encode something about the cytoarchitecture. And now we can ask, what does it encode about cytoarchitecture? And we have multiple ways of evaluating this. One way is to use downstream tasks, and one way is to use embedding methods and just look at, for example, UMAP. So the first thing that we did is we evaluated the learned features on the task of cytoarchitecture classification. So we had a data set of annotated patches from the Jülich brain project, and we trained a linear classifier on our learned features to distinguish between 113 different brain areas. And what we can see here, the little arrow points to the most powerful of the CytoNet models that we trained, is that the models significantly outperform existing automatic approaches. And what we can also see is that if we consider not only top 1 accuracy, but also, for example, the top 2 accuracy, so if the correct area, according to the annotations, is within the top 2 predictions, we reach very, very high performance, up to 92% in brains from which we have seen some training data, and about 75% in brains for which we have no training data. To give some perspectives on this, it's very, very difficult to compare this to actual human performance, because this is very different from how humans approach the task. You would never ask a human to classify brain areas on just these small patches and then ask to make a selection out of 113 areas. So a little bit difficult to compare, but it's fair to say, I think, that this model already performs at superhuman performance when it comes to brain area classification, and with a little bit of post-processing and smoothing of these results, we are quite positive that we can more or less automate the brain mapping process using this method. What's actually, in my opinion, even more exciting is that we can look at these features in a low-dimensional embedding and get an impression of what this model actually learns. So here, what we did is we computed, in this case, 2-dimensional UMAP embedding. The 3-dimensional one is even more expressive, but it's very, very difficult to visualize. This is more of an interactive thing. But what we can see here is the low-dimensional UMAP embedding of features from all the 9 brains that we used, colored by the brain that it originates from. And this is very, very interesting because we see that along the first UMAP dimension and also very, very clearly clustered, we see brain-specific clusters. And this is despite the fact that the model is actually trained to map points even from different brains, as long as they are spatially close in the reference space, to similar feature vectors. So it seems that even though we're telling the model to put certain points, to map certain points to the same feature representations, it still somehow segregates the Latin space. And we're not yet entirely sure why this is happening, but it's certainly interesting to investigate. In addition to this, what we can do is we can, instead of coloring the Latin space by the brain, we can color it according to the maximum probability map of the Jülich brain atlas, which gives us a probabilistic idea of what brain area should be located here without actually knowing the ground truth. And if we do this, we first just see that it's quite colorful without having a very, very large legend. But what we can see is that along somewhat this axis, there are similarities between the clusters. So we have distinct brain-specific clusters, but within the clusters, within the brains, the structure is very, very similar. We can pick one of these brains. I transposed the axis to make it a little bit more layout friendly, and then compare with the maximum probability map of the Jülich brain what we are looking at. And some very, very striking features, without going into too much detail, is that, for example, we can identify this part as being corresponding to a part of the frontal lobe. We also have, in many of the brains, this reoccurring pattern that we have this split in the middle, which is the central sulcus. And we can also identify, for example, parts of the occipital lobe, and we can dig even deeper, which you would then typically do interactively. And it's always interesting to keep in mind that this Latin space is entirely constructed from patches which look like this. So the model at no point has more information than this, and I think this is quite impressive. So there are more things that we can do with this model, for example, layer segmentation, correlation with morphological features, but unfortunately I don't have time to go into detail on this today. The question remains, what's next? So what we want to do next is we want to go, first we want to go beyond the cortex, which is actually right at the moment training on one of the supercomputers, and we already see that it's working even for subcortical and white matter regions. We also want to go beyond modalities, and we have some first indications that this also works. What's also very nice is that the method is not tied to a specific modality. In the sense that we can even train it jointly on different modalities, which of course makes it very interesting for studying, for example, data sets like the extremely brilliant brain that we heard about before. So we already had some discussions on this, and it's also not bound to the species. So applying it in different species like the macaque with the Big Mac data set is also something that we could think of. And finally, I'm sorry, I'm a little bit over time, another modality that we would bring in basically as a cherry on top is textual data, and we discussed this already a little bit yesterday in the panel discussion, basically as an interface to probe the model and give us a way to interact in a way with it in a more natural fashion. So basically, we are moving from this vision of Google Earth for the brain, which we sometimes hear in connection to the Jülich brain atlas, to some kind of a Google assistant for the brain in the maybe not so far future. Thank you very much.